Okay, so it's 3:30, so I guess we better start up again. Right, so just need to finish off the last of the structures in the bacterial cell, and then move on to bacterial identification and phylogeny. So, well, the last structure we need to think about are endospores, which are only formed by some types of gram-positive bacteria. Okay, no endospores in gram-negatives. So the presence and the orientation of an endospore is useful for identification. So bacillus are spore forming, Clostridia are spore forming, and apparently, you can tell the difference between Clostridia and bacillus by the position of the spore within the bacterial cell. So they're called endospores because the spore is formed inside another bacterium. Now, the function of the spore is to have a kind of dormant, very resistant type of cell that can remain viable for a long period of time, even though conditions for growth are very, very poor, and then reactivate, germinate if growth conditions improve. So what is the structure of a bacterial endospore? Well, let's go from the outside to the inside. Okay, so starting from the outside, there may be an exosporium, which is a kind of loosely attached glycoprotein coat, and then a very, very thick layer of spore coat, which is basically packed full of protein. Then you have the cortex, which is a layer of peptidoglycan associated with this unusual molecule called dipicolinic acid, only found in the cortex of an endospore. And then inside, you've got peptidoglycan cell wall of the spore, and then dehydrated cytoplasm with bacterial DNA. So inside this dehydrated cytoplasm, you'll have ribosomes, some DNA repair enzymes, and small acid-soluble proteins, which maintain the DNA in a very condensed state and can protect it from chemical degradation by, for example, reactive oxygen species. Yes? Small acid-soluble proteins. So in fact, they're basic proteins and they bind very tightly to the DNA. Okay. Okay, so the DNA repair enzymes, these small acid-soluble proteins, they are not 100% effective. So if you have a UV radiation, the DNA will get damaged, and these DNA repair enzymes can repair some of it once the cytosol becomes rehydrated. Okay, so what are the functions of these different layers? Okay, so the spore coat, it's just a big, thick, impermeable layer. So, detergents, acid, alkali, they can't really get past this big layer of protein. So it's going to be protecting the dehydrated cell from any kind of chemical attack. Same kind of thing for the cortex. So bacterial endospores are very resistant. You can wash them with alcohol, bleach, leave them for maybe, I don't know, half an hour or something in 70% alcohol. They'll be perfectly happy. Remove it in water, and then they'll remain viable. They can easily resist temperatures of 60, 70, 90 degrees. And that's why, in order to sterilize bacterial media or something like that, you have to heat them at 120 degrees under pressure. That's enough to inactivate bacterial endospores. Boiling at 100 degrees is not enough to totally inactivate endospores. So they're very, very tough. Now, at some point, they have to be able to be reactivated, right? Because there's no point in remaining in an entirely dormant state forever. Just the same, that's just the same thing as being dead, right? You know, what's the point in being a vampire if you never wake up and drink people's blood once every while? So at some point, bacterial endospores have to wake up, they have to germinate, and there are often two steps in this process. So an activation step, which means the endospores got to have some kind of chemical or physical aggression. So if the protein coat starts to get degraded, or if they're heated up to above 60 degrees, this is the activation. Then what happens is there has to be something that also stimulates germination. So that can often be the presence of glucose or amino acids, something like that, which can diffuse past the protein coat because it's been weakened by the activation step and diffuse into the cortex. And this will release a dipicolinic acid. So the dipicolinic acid will come out here, diffuse back into the spore coat. Some of the proteins in here are enzymes. The dipicolinic acid will activate them, and these enzymes will break down the spore coat and the cortex. And that's what's going to allow the cytosol here to get rehydrated, because now water can come in, rehydrate this tiny cell inside the endospore, and then it will start growing and produce, vegetatively growing active bacterial cells. Okay, so two steps, activation and then germination. So what does that mean? Like, it means that, okay, the bacteria, the endospore will only germinate really if it's in the presence of favorable growth conditions, because there's got to be something like amino acids or sugar locally in order to stimulate the germination. So they only wake up, when there's something to eat, okay? Okay, so that's the function of bacterial endospores. How do they get produced? Well, you've got a bit of a problem here, because the endospore itself is totally metabolically inactive. It's not producing any proteins, it's not doing anything, so how can it produce all of this like big thick spore coat and a peptidoglycan layer here? Well, the answer is it's not produced by the endospore, but produced by the other cell. So sporulation is a process that, well, generally speaking, it's induced when bacteria don't have enough to eat anymore, okay? So if nutrients are limited, then spore-forming bacteria will stop growing and start to produce endospores. That's not the only stimulus for sporulation. It can be some other physiologically relevant signal for that bacterial life cycle. So for example, Bacillus anthracis, the anthrax bacillus, will sporulate when it's in contact with high concentrations of oxygen. Why is that important? Well, this is a bacterial pathogen which infects and kills livestock, and when animals are infected, they'll bleed, they'll spit out blood which contains a lot of anthrax bacilli. So when these anthrax bacilli come out of the animal and end up lying on the ground in the field, they are not in the correct conditions for growth. Okay, they're not inside the infected animal anymore. They are in contact with higher oxygen concentration just from the surrounding air, and that's what induces sporulation. So they produce spores, and these spores remain in the ground. So the induction of sporulation by contact of oxygen, that's why when you've got a suspected case of anthrax in livestock, in cattle, the vet can't come in and start like cutting the carcass up in the field because that's dangerous. It's going to contaminate the field, okay? The whole animal's got to be taken away to a laboratory, and the lab work is done, you know, in the laboratory. Okay, so that's just a bit of a context here. So what happens when sporulation occurs? Okay, so for rapidly dividing bacterial cells, you get binary fission, right? So the bacterium grows a bit bigger, then it splits off into two daughter cells which are identical, and each of these daughter cells will also divide. During sporulation, what happens is the cell division is going to be asymmetric. So you have one daughter cell, which is smaller than the other one, and this will become the endospore, and one of the daughter cells, which is bigger, which will become the sporangium, okay, the structure that supports the spore. So the larger cell will start to engulf the smaller one, and the smaller one is going to shut down all of its metabolism and desiccate its cytoplasm. So it becomes metabolically inactive. Then the larger cell, the sporangium, will start to synthesize all the layers that we saw in the endospore. First, the peptidoglycan cortex, then the spore coat of protein, and then the exosporium. So at the end of this process, you've got an endospore, which is metabolically inactive, surrounded by all these layers that have been produced by the other cell. And then finally, the sporangium is going to kill itself. It's going to die and lyse and release just the free endospore. Now, why is this interesting? I know that's the question you're all asking, right? Why is he telling us about this stuff? Okay, from a bacterial point of view, okay, for spore-forming bacteria, it's a way to, you know, survive conditions which are not favorable for growth, okay? So that's why these bacteria do this. They produce endospores, the endospores are resistant, and they'll grow back when conditions are better. So that makes sense. But from a fundamental biology point of view, this is very interesting because you've got two fundamental processes going on here. One of them is cell differentiation. Normally, when we think about bacteria, we're thinking about, okay, they grow, each bacterial cell, they're all identical to each other, they just make identical copies. That's not what happens here. You have this cell division, and you have two cells which are going to perform different kind of genetic programs and have two very different cell fates, okay? This one is going to die, and this one is going to become an endospore. It's very simple, but this is the beginning of cell differentiation to two different cell types. The other process that's going on here that's also very interesting is programmed cell death. This cell here at the end is going to die. So these two things, they're fundamental processes for the differentiation and development of multicellular organisms. During embryonic development, differentiation and programmed cell death are absolutely essential. So normally we think of, you know, these processes are intrinsic to complex multicellular organisms like us, but in fact, they already exist at a very, very basic level in bacteria, okay? So that's a lesson here for spore formation. So, you know, we have to try and understand how this works at a molecular level to try and figure out what are the bases of cell differentiation in prokaryotes. Okay, so, all right, let's see if you've figured this out. So here's a little question about sporulation. And it, you know, it concerns everybody's favorite meal, right? Hang on, I have to open the question first. Yeah.
When it's taken boiling point, does it mean 100 Celsius degree?
Yes, boiling point for pure water at 1 atmosphere pressure, if you want to be really precise about it. Whew. I never had that question ever before.
Because you gave us a special temperature point for killing bacteria.
Oh yeah, that's 120 at high pressure. 20 minutes at 120 degrees is required to inactivate endospores. So it's less than that. Okay, 23 replies. Is that okay for everyone? No? Think about it. $25. Yeah, well, I think that's enough of that dangerous thinking. You know, don't want to have too much. Who knows what would happen next, huh? Okay, so here's the answers. That's what people think. They're killed during the first heating cycle. That's wrong. Okay, so nobody chose that one. That's good. 2. Well, I thought this was believable. It's not the right answer, but I try and make a believable false answer. So yeah, that's also not true. So you didn't get this one. Most people think they survived the entire process, but that's wrong. That's wrong. 3 is the correct answer. Okay. What's happening here? Okay, right. So 100 degrees, it's not enough to inactivate them. It's rather the opposite. It's enough to activate the endospore, right? They'll get like heat damage to the protein spore coat. Then, why are they going to germinate? They're in a tin of casserole, right? There's a very, very rich culture medium for bacteria. They're going to love it. Oh, they're going to love that casserole. They're going to love those beans, man. They're going to love it. So you have, amino acids in there. They can diffuse across the spore coat, activate germination. That's what's going to happen after the first cycle. Once they germinate, they are going to become vegetatively dividing, actively growing cells. Those cells are going to be killed because they're not heat resistant. Okay?
They are not, what did you say?
Heat resistant, because they are not endospores anymore. They're just regular dividing bacterial cells. So the first cycle activates the endospores, they germinate, and they become actively dividing cells, and then the second and the third heating cycles will kill them. Okay, so that's what happens with this process. So it's not used very much anymore, but it used to be used like in the 19th century to prepare, tinned food. They would be heated like 3 times in a row in order to ensure that no endospores survived. Yes? What do I mean by activated? I mean, when I was on this slide, I said that endospores are very resistant, but at some point they have to become reactivated and germinate. That's what I mean, okay? So there are often two signals: activation either by heating or chemical attack on the protein coat here, and then germination, which is dependent on the presence of amino acids or sugars that can diffuse in and cause the release of the dipicolinic acid. So activation is the damage to the spore coat. Well, they're related, but okay, so if you put an endospore into a rich medium and don't do anything, sometimes they won't grow. Okay, sometimes you need to do something else to them first. All right, and that can be heating them up a little bit to damage the spore coat. Yes, allow germination. Yeah. And in our example with the tin of cassoulet, we have both, right? We have the heating for the activation, and we've got the, nutritionally rich environment which induces the germination afterwards. Yeah, yeah. Okay. So that's what's happening here. Okay, activation. Okay. Right, so we have, about an hour, I suppose, to think about identification and phylogeny taxonomy for bacteria. Now, thinking about phylogenetics and classification, there's a kind of historical context to this, in that microbiologists started to, classify bacteria before molecular biology had been invented. Okay, so the first attempts at, grouping different bacterial species together into genera, different genera together into families, was all based on phenotypic or phonetic classification. And ever since DNA sequencing has been invented, then this has kind of been replaced, or has had to be kind of, let's say, the phonetic classification has had to be altered based on molecular phylogeny. That is, how close genomic DNA sequences are to each other. And currently, the classification system for bacteria is a kind of a mix of these two systems. So the older phonetic classification system, and then with elements of molecular biology, molecular phylogeny, that kind of changes things sometimes. So the reference work on this is Berge's Manual of Systematic Bacteriology, which must be in its, 500th edition by now. And so the idea behind this was to try and classify all bacteria in a kind of Linnaean style of classification like you have for animals and plants, okay? So you've got 1, 2, 3, 4, 5, 6, 7 levels, 7 taxonomic levels in the classification: species, genus, family, order, class, phylum, kingdom. So for example, Shigella dysenteriae, that's the species name. Just like for any other species, the first name in the binomial classification is the genus, and then several genera. It's in the family Enterobacteriaceae, order Enterobacteriales, gamma Proteobacteria, Protobacteria, bacteria. So, okay, family always ends up with ace somewhere, and order ends up with ale. Right? I go to a bar and I order an ale. And that's how I remember it anyway. And my family is ace. Okay, so the idea is we're trying to get bacteria into these 7 taxonomic levels, just like the rest of biology. Now, the kind of problem microbiologists had here, right, is if you want to have lots of different types of classification, you need to have a lot of phenotypic characteristics in order to differentiate different species. So what kind of phenotypic characters can we use for bacteria? Well, the first one is just basic morphology, okay? Gram plus, gram minus, bacilli, cocci. Sometimes you can even use colony morphology, whether they've got an endospore, where the endospore is in the cell, this kind of stuff, whether flagella are present or absent. So you've got this kind of morphological characters, but there's not a lot of detail in there, okay? So you've got two let's say, groups here, 2 there. So you've got like 4 possibilities here. Flagella or not, you've got 8 possibilities. Endospore, yes or no, okay, 16 possibilities. You know, that's not really a lot, okay? So you need to have some more characters in order to be able to distinguish more and more different types of bacteria. So what are they? Often they're based on biochemistry. That is, you know, what carbon and nitrogen sources can be used, whether they're aerobic, anaerobic, this kind of stuff. What's their general nutritional type, photosynthetic or not. What their growth characteristics are, whether they can grow at low pH, what's their optimum temperature range, this kind of stuff. So these are a bunch of biochemical characteristics that can be measured for different bacteria, and it gives you kind of that's what their phenotype is, okay? And another thing you can use is, you know, whether they infect different types of species, if they're pathogenic, or whether they are symbiotic or just free living. So that's what a phenotypic description of bacteria is, right? Some morphological characters, but most of them are biochemical. And what this is useful for is identifying bacteria, or at least having a few simple criteria to kind of orient the test that you need to do to identify something. So an application of this phonetic approach is to have a kind of identification key. So if you've got an isolated colony on a Petri dish, the first thing you want to do is, you know, perform a gram stain, and then you'll find out whether you've got bacilli gram-positive or gram-negative bacilli and cocci, and then perhaps perform some simple biochemical tests. Are they catalase positive or negative? So, for example, if I've got some gram-positive cocci which are catalase positive, well, they might be Staphylococcus, right? If I've got gram-negative bacilli that are oxidase negative, they might be Enterobacteria. So these morphological and simple biochemical tests give you some kind of idea of what you're dealing with, but they're not enough to identify bacteria down to the species level. Now, to do that, you have to use the results of the orientation test and then do something else. So in the lab class, what you're going to do is set up an API gallery. So an API gallery is a set of biochemical tests, okay? To test whether bacteria can use particular sugars to grow, okay? So here, okay, this one can use glucose, can ferment mannitol, cannot ferment inositol. So for each of these little tests, you've got a result, positive or negative, and that tells you something about the phenotype of the bacterium, and you can use that for identification. Okay, so first thing, you start with an isolated colony on a Petri dish. Why is that important? Because a colony started from one single bacterium, and then it grows, so every bacterium in that colony is exactly the same as all the other ones. So you're looking at one species, one strain. Next thing is that, okay, the orientation test, you can do that, you know, quite quickly, one day. But this kind of test, what you need to do to identify a bacterium down to the species level, will take one more day. You have to put them in culture again and check it the next day. So that is a bit of a limitation, and that's why I kind of, the other thing I need to say about this is that, okay, so you get a kind of like tree structure coming out of this identification key, but don't forget, this is not a phylogenetic classification. It's just a kind of guide for what test you need to do in the lab to have an idea of what kind of bacteria are present. But it doesn't mean that, for example, Staphylococcus and Streptococcus are phylogenetically close together. Okay, so another thing that can be done to identify bacteria rapidly is MALDI-TOF mass spectrometry. Anybody know what that means? Yeah, but the MALDI-TOF bit.
Maybe the brand.
Huh?
Maybe the brand.
What?
Maybe the brand. I'm kidding. I'm not sure.
Okay, TOF, that's time of flight. Okay. Yeah, this is matrix-assisted laser desorption ionization time of flight. That's what it means. Okay, so let's try and figure out what that means. So what you do is you take a little bit of your bacterial colony and you scrape it onto the MALDI plate. There's a kind of metal plate, it's got a place for different samples. So you've got the plate here with your sample, and then the laser is going to zap one of the spots where you've put the bacteria. So that's the laser desorption. So the laser is going to zap it, got a lot of energy there, so all of the proteins and macromolecules inside that are in the bacteria are going to be desorbed from the matrix plate. They're going to go up into the gas phase, and they'll be ionized, so they become positively charged. And then they get shot through this hole here, the charge grid, and then they fly off, and then they get detected. So the mass charge ratio is going to determine how fast they fly or something. Okay, it's physics, all right? Don't ask me about it. All right? So, and then you get the mass spec reading here. So, you've got some kind of spectrum which depends on what? It depends on basically what is the overall protein macromolecule content of this particular bacterial species. How do you get from this to an identification? Well, the companies that make these machines, they have run hundreds and thousands of strains of bacteria through this machine. So basically, they have a big database of what the spectrum looks like for Escherichia coli, what the spectrum looks like for Pseudomonas aeruginosa, what the spectrum looks like for Salmonella. And so you match the spectrum you have for your sample to the spectrum in the database, and then you get the identification. Now, that's kind of pretty complicated. Oh, that's just what I said. But the big advantage here is, one sample can be identified to the species or subspecies level in 30 to 40 minutes. And with, a modern machine, you can run 200 samples in a day. So you get the identification in the same day. And that's very important, especially in clinical microbiology, because if you've got some kind of pathogenic bacterium that's on a plate, it might be very important to identify what it is in order to use the right kinds of antibiotics. And if you have to wait one day before you get the identification, that could have a big impact on the patient who's very, very sick. So it's very useful to have identification same day through MALDI-TOF. So this is kind of, there's a machine like this in the bacteriology lab in the Nantes hospital, for example. So that's kind of more modern technique for identification. Okay, yeah, so let's have a little question here so I can have another break. Let's stop. I haven't activated the question yet. I have to kind of go back up here. Yes.
By positive identification, does it mean gram?
Oh yeah. No, no. I mean you have to. Okay. What I mean is, if you perform a gram stain and you get it's a gram negative, okay, it's not that. But positive identification means yes, it's really that. So that's why I have positive in the sentence there. But it's got nothing to do with gram positive, gram negative. Okay. So if negative identification, can you exclude? How can you be sure it's not? Da, 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 da. But that would be a different question. Everybody ready? Whoa! Wow! There. See, everybody heard you, and I had to see the numbers changed since then. Oh, hang on. Can you take your votes back because the numbers are going up and down? Wow. Okay, so let's have a look. All right, so here we go. Number 4, right? I don't recommend this technique, okay, because they probably don't know any better than you do. Even if you think, oh, this is the genius girl in the class, I better ask her. Well, you know, she's probably not that great, right? Okay, so yeah, that's right. This is the correct answer. This is wrong because, okay, you might get Staphylococcus from this, but you need a species level identification. Orientation tests, gram stain, catalase, oxidase, this kind of stuff. They are orientation tests. They're not enough to get the species level identification. So that's why 3 is the correct answer, because you need to know if it's Staphylococcus aureus and not Staphylococcus something else. Okay, so that was the point of that question there. Yeah. Okay. Oh, my animation's all screwed up here. That's not good, is it? Okay, so I guess I better just do this before, right? So that's species level identification, okay? You start with a pure colony on an agar plate. You run orientation tests and then some other biochemical tests to get the species level identification. An API gallery or run your MALDI-TOF machine, and then you get species level identification. Now, in many cases, it's interesting and important to get a subspecies level identification. Which strain of Escherichia coli or which strain of Streptococcus pneumoniae have you got? For Streptococcus pneumoniae, very many strains are not invasive and do not present a danger to the person who's carrying them in their nose. But some strains are invasive and can cause meningitis. So you want to be able to determine whether you've got a pathogenic strain of Streptococcus pneumoniae or Escherichia coli in the person that you're dealing with on your agar plate. So that's why subspecies identification is very important. Now, there are different types of approaches. Some of them can be biochemical, you know, whether your particular strain can ferment a particular sugar or not. But perhaps the most frequent types of tests that are run for subspecies identification are antigenic. So you want to determine what are the K, H, and O antigens on this particular strain. And you might want to find out what is the genotype. So if you run these antigenic tests, you'll determine the serotype. But for other reasons, or complementary, or in addition to that, it might also be interesting to determine the genotype of this particular strain in order to find out, for example, if this strain has a gene coding for particular virulence factors. So how will you do this? Well, you can run a PCR reaction with primers specific for the gene you're interested in, and you'll get a result that is positive or negative, okay? So you combine these two things together, and then you have a description of the particular bacterial strain. So your Escherichia coli, this is a species level identification, 0157H7, so that's its serotype, right? And then it carries the STX2 gene. So this is Shiga toxin 2. And that's the genotype. So genotype, it's always written in italics, okay? So those are the techniques used for subspecies identification. Okay, so that's identification. How about, oh yeah, so what I'm going to say now, yeah, forget because I can't really see the slides here. Yeah, so serological tests, how do they work? Well, the most common one is the agglutination test. So what you do is, once again, you need to start with an isolated colony on an agar plate. You pick a little bit off your colony, mix it up with some water or saline solution, and then just mix it up with some latex beads that are conjugated with antibodies specific for a particular antigen. So for example, you might be talking about antibodies specific for the O157 O repeat in the LPS. So you mix them up. You mix 1 drop of latex beads with bacteria from an isolated colony and incubate for 1 minute at room temperature. So if the antigen is not present on the bacteria, the latex beads are not going to bind to the bacteria, and they all stay in solution, in suspension rather. So there's no interaction. So this strain will be O157 negative. The O157 antigen is not present. However, if O157 is present, then the antibodies on these beads will bind to the LPS on the surface of the bacteria, and then they will all kind of agglutinate together. Yes?
I don't understand why we have to eat with heat, no? We shouldn't
No.
after having the...
No. RT, room temperature.
Okay.
It's not heating.
Okay.
So it's just on the bench, put the beads in there, 1 minute, and then you look. Okay, you get a visual response. So if the bacteria and the beads are just like floating around in suspension, you see something like this. That's a negative result. If they agglutinate, you can see they form kind of like clumps just on the kind of like paper strip or the plastic strip where they are. Okay, so you can see it just with your eyes. And you have to look after just like 1 minute. If you wait for too long, you know, they could just like clump together and it's not specific. Okay. Okay, so that's how this works. Very convenient, very easy to do. It's fast, right? Okay, so that's how an agglutination test works. Okay, so that's identification, orientation test, some other kind of set of biochemical tests to get species level definition, serological and genotyping to get subspecies identification. Now the rest of it's okay. The talk for this afternoon is about taxonomy. So first approaches here were to try and use phenotypic characters, you know, gram positive, gram negative, all these biochemical tests to try and group together bacteria that are very, very close phenotypically and try and derive phylogenetic trees from this information. So what you can do in order to get this is, you know, score each character as present or absent. Okay, so ferment glucose, yes, okay, then that's 1 for glucose fermentation, something like that. Okay, ferments lactose, no, 0 for lactose. That's the kind of idea here. And then you compare each pair of bacterial species. So for each species, you've measured a whole bunch of characteristics. They score 1 and 0 for each one. Then you take them pair by pair, and you try and calculate the matching coefficient. So you've got a kind of table here. So both bacteria can ferment glucose. That's one character that's in here. This bacterium Y ferments lactose, but bacterium X does not ferment lactose, so that's one character that goes in this cell of the table here. Okay, so you count up the number of characters that are corresponding to these different situations, and then you can calculate a matching coefficient to tell you how close the bacteria are to each other in phenotypic terms. So, simple matching coefficient is, okay, characters that are shared by both species of bacteria and the characters that are absent from both species of bacteria divided by the total number of characters you have looked at. So that's a simple matching coefficient. This might not be so good because is it really relevant to count things that are absent? So you might get some false results, you know, including characters that are absent from both species, and you probably don't want to do that. So probably the Jacquard coefficient is more useful. So you only count the characters that are shared, and you divide it by the characters that are present in at least one of the two bacteria. Now, this coefficient is going to be somewhere between 1 and 0. Imagine your bacteria, they have nothing in common. So like A, this is going to be 0 here, right? And B and C will be some kind of number. So if they are totally different, the matching coefficient and the Jacquard coefficient, they're going to be 0. If they're absolutely identical, it will be 1. Because you'll have, you know, so if you've got 50 characters here, you'll have 50 there, 0, 0, 0 here. They're totally identical. Or it might be like 50, 0, 0, 10. Something like that. Okay, so totally identical is 1, totally different is 0. So you'll have a bunch of numbers for each comparison for each pair of bacteria between 1 and 0. So that's what gives you your similarity matrix, which I've illustrated here. So for each pair of bacteria, there's a number here between 1 and 0. And once you've got that, then you can use distance methods in phylogeny to give you a phylogram, right? Phylogenetic tree. So people who were in L1 here, you should know about this, right? People who were not in L1 here, do you know what I'm talking about? Okay, no. Anyone else? Don't know how to do this stuff. Okay. Well, the basic idea is, okay, what you want to do is you want to group together species that are closest phenotypically. So here, right, species 1 and species 2, their similarity index is 0.92. Okay, 1 is totally identical, 0 is totally different. And these are the two closest species. So you put them together in a group like this. And then the algorithm does it for you, all right? Like you just like put the numbers in, you press start, and it will produce a tree for you, right? And then it's going to group together the different species according to how close they are together in this similarity matrix. So you can see, you know, 5 and 6, they're kind of close together, but different from most of the other ones. 4 is different from everyone else. If you look at the numbers here with 4, they're all quite low, right? So that's why 4 is on this branch further away from the others. But it's not really my objective to redo molecular phylogenetic approaches for everybody, I'm afraid. So, but that's the basic idea, right? So you could use WPGMA or neighbor joining to work out the tree from this kind of data. Okay. So that's what some people tried to do. Now you have to go back quite a long way to find a publication where somebody is actually trying to use this stuff. So here is the data, and here is the phylogenetic tree that came out of a publication from 1994 where the microbiologists were trying to classify different types of Neisseria. Okay, Neisseria gram-negative cookie. They include Neisseria gonorrhea, Neisseria meningitidis, important pathogens, and a whole bunch of other Neisseria which are harmless. Okay. Now the problem with all this phenotypic approach is you need, if you want to classify 100 strains of bacteria, you need to weigh a lot of phenotypic characters. So this is the table they have here, right? So these are all the different strains of bacteria that they looked at, and these are all the different tests that they performed on all of them. So they got all these results, and they put them into the algorithm, and you get this kind of phylogenetic tree. Whew. So what's the result? Okay, so in fact, it looks like Neisseria gonorrhea, the two groups, they go together, and they're distinct from all the other ones. Within the pathogenic bacteria, meningitidis, gonorrhea, they're kind of in this clade that also includes Sneath. This didn't work though, right? Look, they've got a problem here. They've got Neisseria meningitidis group B, Neisseria meningitidis group A, and then they've got this other thing in the middle. Whereas normally you would think these two would be together, right? So this is looking a bit strange. But maybe it's right, okay? So that's the result from 1994. Good effort from Barrett and Sneath there, I think. Right, what else can we say about that? So that's, that's how to get classification, phylogenetic classification from phenotypic characters. Difficult, but not impossible. So this has largely been replaced now by molecular techniques. So I'm just going to say a little bit about this table because different types of molecular technique are useful at different levels of the classification. So the first thing that people did, even before DNA sequencing was invented, in the 1960s and the early 70s, was they tried to measure the general overall genetic similarity between two species by genome hybridization. So what does that mean? That means you've got two bacterial strains. You've got A and you've got B. So you take the DNA from species A, you put it on a membrane, and you take the DNA from bacterial species B, you label it radioactively, you chop it up into small fragments, and then you hybridize it onto the membrane. And you measure how much radioactivity you have in total here. You hybridize it onto the DNA from bacterial species A, and you wash it off, and you see how much has been hybridized onto the membrane. So if these two species are identical, their DNA is identical, right? So you'll have 100% hybridization. If they are totally different, their DNA sequences will be very different. You'll have a very low level of hybridization. So this is a simple quantitative measurement, and you can actually classify different genera and different species of bacteria this way. It doesn't really work for subspecies, and it doesn't work for higher level classification. So since then, DNA sequencing was invented, and you can sequence different parts of the bacterial genome. One thing that's very useful is 16S ribosomal RNA sequencing, or the genes that code for the RNA. And that's very useful for higher level classification up to genera and maybe species. Multiple locus sequence typing, what is that? You choose several genes to amplify up by PCR, and then you compare their sequences. So that can be useful for subspecies, species, genus, maybe family level classification. And more, you can sequence whole genomes and try and classify bacteria by whole genome sequencing. So I'll give you kind of one example of that. So this was an article, 20 years later after the phenotypic classification of Neisseria species. This is a publication with classification of the same types of bacteria, this time with a molecular genetics, kind of molecular phylogeny approach, using the sequences of 53 ribosomal subunit proteins. So this is multiple locus sequence typing, that's MLST. You don't just sequence one gene, you sequence several genes, and you put them all together and you get the overall similarity. So that's based on the sequence of 53 different genes here. So what do you get here? Okay, you've got the gonorrhea group is there, meningitis group is here, the polysaccharia group is there, and the Sneath group is over here. So in fact, this result there, the one that looked strange, is actually wrong, because these Neisseria cinerea are not genetically similar to the meningitis at all. Okay, so this is an example of, you know, the molecular phylogenetic approach is definitely more powerful than this one. Okay, and because sequencing, you know, is very cheap now, so there's basically no limit on how much stuff you can sequence, whereas all this phenotypic approach, that took a lot of time and a lot of different tests. Okay, so that pretty much works, okay? So that's pretty good for classifying different species within a genus. Okay, so we know which ones are closer to which ones. So how about using it to classify all bacteria? Is this going to work? So here's a paper where they got different bacterial strains from the class Bacilli of the phylum Firmicutes, which are the low GC gram-positive bacteria, and did the same kind of thing, sequenced the 53 ribosomal protein genes from all these different bacteria and used, you know, neighbor joining or whatever to to classify them. So that doesn't look so bad, because, you know, you've got like all the Streptococcus, they're all grouping together in a clade, all the Lactobacillus, they're all together. One of the interesting things is that, in fact, Staphylococcus is not close to Streptococcus at all. Actually, they are really pretty different, these types of bacteria, even though if you look at them in the microscope, they look very similar. But phylogenetically, they're quite different. The only thing that, you know, something's wrong here, though, right? Can you see something wrong? Yeah.
You know, why the 13 of Bacillus is just too far away from the 14 one.
Yeah. So this seems to be wrong. So it's not really working 100% correctly. I don't know why. I didn't read the paper. I just like picked out the figure. So mostly it works, but, you know, it's not 100% correct. But basically, that's kind of okay. It kind of fits, you know, things are where they should be. So within a family, that seems to be okay, right? So how about like at a kind of larger, higher level scale? So here is what they did in that paper. All prokaryotes, okay, all eubacterial groups. Now, in fact, there are several inconsistencies here, and I would like you to take a little look and tell me which groups have problems. Whoa. Okay. Non, ça marche pas. Comment ça? Comment ça marche pas? It should work, I promise, I promise. Wait, wait. Normal. In case I like copied the wrong code or something. Should be the right one. That's the same, right? Yeah, it should work. Yeah, but, okay, right. Try now. Yeah, okay. Yeah. Okay. Everybody okay with that? Yeah. Yeah, I think you're pretty good answers here. Okay, good. Yeah, okay, so let's see what people think. So, okay, that's Bacillus, that seems okay, Bacilli. Actinobacteria, a couple of people said that. Clostridia, Proteobacteria, those are the ones with problems, right? So, yeah, and I think most people saw this one, Clostridia. Most of them are here, some of them are down there. They should all be clustered together, right, if this is working. And the Proteobacteria, this is a big phylum of gram-negatives. So you want all the Proteobacteria to be together, but you've got the beta and the gamma are here, and then you've got like the delta and the epsilon over there. Okay, so they're all over the place. It's not working at all. So there are problems. There are problems with this approach. Okay. Okay. So that's multiple locus sequence typing. And the other commonly used technique for higher level classification phylogeny of bacteria is using 16S. What am I doing? It's not. Yeah, 16S rRNA sequencing. You can identify different species and different higher level groups. So the idea is that most bacteria have one copy of the ribosomal RNA operon per genome. That's not always true, in fact. And within the gene coding for the 16S ribosomal RNA, there are regions that are highly conserved, and there are regions that are variable. So, if you choose primers that are in the constant regions, that are in blue here, they can hybridize to the DNA of pretty much every bacterial species. So you amplify something up. And then, for example, here you've got this primer in a constant region, this primer in a constant region. In the amplicon, you'll have a variable region. And you sequence the variable regions and you use that for your classification. So the idea is that, you know, you line them up, you see where you've got differences, you count the number of differences, and then classify bacteria according to how identical they are. So this was proposed, you know, 25 years ago or so, as a general technique to determine whether two bacterial strains belong to the same species or not. So at that time they said, okay, if you've got more than 97% identity, then these two bacterial strains, they belong to the same species. So I chased this up, and so, you know, 10 years later they revised that, their estimate with more data. So in fact, now they say 98.7% identity, they're the same species. Less than that, they're not the same species. Okay, so just from sequencing one gene, you can get a general idea of how close or how different two bacteria are. So that's pretty useful. On the other hand, you might think, okay, what happens if you've got 98.65% identity? You're kind of in trouble, right? Okay, so does this really tell us something interesting about higher level classification of bacteria? So this is a phylogenetic tree I copied out of the textbook from Prescott's Microbiology. So it's a simplified phylogeny of the major groups of bacteria, eubacteria and archaebacteria, based on 16S ribosomal RNA sequencing. And what is quite reassuring is that it kind of makes sense, okay? Because you've got all the gram-negatives grouping together in a clade, and you've got the classical gram-positives grouping together in a clade here. Okay, the high GC gram-positives and the low GC gram-positives. So these are two sister groups, okay? Gram-positive, gram-negative. Another thing that's kind of interesting about this is that, you know, the basal groups here that are very close to the branch point with archaea, Aquifex and Thermatoga, they are both hyperthermophiles, thermophilic bacteria that grow in extreme environments. And many archaea also grow in extreme environments. So you might think, oh yeah, that's great. That's telling us that the origin of life is in some kind of like extreme hot springs or something. So that's also quite interesting. However, I think I'm going to talk about something that means that that might not actually be true in a minute. So one of the other questions you can ask about this is, what was the original state, okay? Gram-positive, gram-negative. You know, in phylogeny, you talk about the apomorphy, plesiomorphy, you know, the ancestral state and the derived state. Okay, so for the bacterial cell wall, what was the ancestral state? Gram-positive or gram-negative? So in order to know that, you can't really tell from here, but you'll have these as the outgroup, right? So they can kind of let you know what was the ancestral state of this characteristic. So what is the gram status of these different guys here? So Deinococcus thermus, they have an outer membrane, but they don't have LPS, so it's not really gram-negative. Chloroflexi, they're gram-positive. Thermatoga, just let me check on my cheat sheet here. Oh, I can't, okay. I think they are gram-positive, but Aquifex is gram-negative. So the whole thing is kind of screwed up. So how can that possibly be true? So if you look in the genome of Aquifex, there's quite a lot of the genome sequences that are derived from proteobacteria. And if you look in the genome of Thermatoga, there is a significant percentage of the genome that comes from archaebacteria. So they are not really strict lineages, okay, Aquifex and Thermatoga. They are kind of hybrid bacterial species that have been affected quite strongly by lateral gene transfer, exchanges of large amounts of DNA between very different species of bacteria. So that is true for these two groups of bacteria, and that means that it's very, very difficult to interpret anything about their evolutionary history, okay? So it might just mean that these two guys are at the base of the tree, but it might just be because they're very difficult to classify because their genomes are all over the place. So it's possible that, okay, they are hyperthermophiles, but maybe their phylogenetic position here just doesn't mean anything. So we can't really overinterpret this, or what I said before, or maybe the origin of life is in some kind of hot springs. That's going too far because phylogenetically, these guys are totally screwed up. Okay, you can't really tell anything about them. But you know, up until that point, I think it's okay, and probably the ancestral trait was gram-positive. I think most microbiologists would agree about that, and that the external cell membrane is an innovation, and that's a derived state. Okay, so what else is very interesting about 16S ribosomal RNA is that one of the very important findings from Carl Woese, who first did this in the 1970s, was the observation that 16S, or short chain, or the small subunit of the ribosomal RNA sequences, are very different between eubacteria, archaebacteria, and eukaryotes. And this led to what is known as the three-domain model for for life. And this was a big thing that's had a major impact on thinking about the evolution of life. So that means that Carl Woese said it doesn't really make any sense to group together archaebacteria and the eubacteria in one group, because in fact we should be thinking about three domains, not eukaryotes, prokaryotes, two domains, but eukaryotes, archaebacteria, and eubacteria. So that's a big difference here, big change, and that really kind of calls into question whether prokaryote, does that have any meaning? Well, personally, I think that prokaryote does have a meaning, and most microbiologists think that way too, because these are very simple cells that live in the same kind of way, and their biology is similar enough for the group prokaryotes to make sense. The other thing that's interesting about this is, this is a phylogram, right? So the branch length is supposed to be related to the time between the different events in history. So if you look at the eukaryotes here, the branch length here is really long. So that means if this is an accurate reflection of history, that must mean that eukaryotes are really ancient. But there's no really good microfossil evidence for eukaryotes being around, a billion years ago, or maybe 2 billion years ago. But they're not as ancient as archaea or bacteria in terms of the microfossil record. So we can kind of ask, does this really represent the history of life, or is there something wrong with it? And one of the things that is wrong with this in its original form is that, okay, we knew a lot of eubacteria, we knew a lot of eukaryotic organisms, but we didn't really have a lot of archaea to compare with. So the shape of the tree might be misleading because we don't have a good sampling of this clade here. So that's something that has really moved a lot. So in 2015, there was a very important paper about the description of the genome sequence of a new type of archaebacteria, the Loci archaeota. And so they had 3 genome sequences formed from shotgun sequencing, and then they match up the contigs to get a whole genome. And what is interesting here is that when you put the sequence of 36 conserved proteins, so this is MLST, and kind of determine the phylogenetics, you've got all of these guys are archaebacteria, eukaryotes are very close to these loci archaeota. So with more sampling, you don't really see eukaryotes as a third domain of life. You see them as a subgroup of the archaea. And that's very, very interesting because, you know the endosymbiotic theory for the origin of eukaryotes, the mitochondria, they come from some kind of proteobacterium. So what was the other partner, okay? This is very interesting because it's telling us that it's something close to the loci archaeota, something up here. That's the other partner in the symbiosis. And the other thing that's interesting about them is when you look at the type of genes they have in loci archaeota, you've got a lot of these things that were considered to be unique to eukaryotes and had never been found in any other type of bacteria, eubacteria or archaebacteria. Things like proteins that are involved in membrane transport, or these escort proteins not present in any other archaebacteria, GTPases, this kind of stuff, they're present in this loci archaeota. So that was a really big story, it was a really big deal. However, you could say, well, this is just fragments of DNA down from the ocean underneath the Arctic Ocean off the coast of Norway. It's not actually a real organism, they just screwed it up. They've got their DNA sequences and they stuck them together, but this isn't a real thing. So just last year, there was a publication, and it was in preprint last year, but it's been published in Nature this year, where they actually managed to culture some of these things. And this was an amazing piece of work. So they got some kind of like sludge from the bottom of the ocean and then tried to grow it up in culture. There was a lag phase, 1 to 2 months in culture, nothing happens, doesn't grow. And then when it started to grow, the doubling time is about 2 to 3 weeks. Now, normally in the lab, we work with E. coli, Staphylococcus, something like this. Their doubling time is 30 minutes or 1 hour. So you put it in the culture, in the lab, in the incubator one night. The next morning is full of bacteria. Look at this. Whoa, it takes like 6 months to do one experiment. So the whole project took them 12 years. But in the end, they managed to culture these bacteria and identify them. So that's really fantastic. So they got the sequence. These are real loci archaeota. So it's a real thing, all right? This is real bacteria. And what is interesting is they have this kind of weird like projections here. They don't have any organelles, but they've got these kind of like filamentous membranous projections. And they live as symbiotes with other bacteria. So just in 2 minutes, and that's the end, then we have these results have given us really a new model for the origin of eukaryotes. Okay, so the idea is that you've got one of these ancestor loci archaeota that's living with a methanotroph or sulfur bacteria. Then when you start to get more oxygen in the atmosphere, then they start to get involved in a symbiosis with proteobacteria. Then little by little, they kind of internalize them, and then that becomes a eukaryotic cell. So I think this is a big kind of deal that's just come out into the open in the last 5 years or so, these new types of archaebacteria that are the missing link in the endosymbiotic theory. Last slide here. What are the problems in bacterial taxonomy? The definition of bacterial species. You know, you have different molecular phylogenetic definitions of what constitutes bacteria within the same species. These numbers, you always wonder whether they aren't a little bit arbitrary. What do you do with something that's 70.01% hybridization or 98.72% identical? It's very difficult. Sometimes you have inconsistencies with traditional species genus names. So Shigella and Escherichia, these should be two different genera, right? But in fact, they're very, very similar. Basically, they're the same species here. Clostridium difficile, important pathogen in hospitals. Actually, it's not really Clostridium, so it doesn't group with Clostridia. So then the people who did molecular phylogeny said, yeah, we should change the name of this thing and call it Peptoclostridium difficile. Then all the medical microbiologists said, what, are you mad? Everyone knows this is C. difficile, right? So they had to like change that, and now they're called Clostridioides difficile. So now everybody can still, if they've got C. difficile infection, that's okay. And really last slide. The main problem is down at the basal layers of the phylogeny here, events that happened really a long time ago. We always try and interpret this as a kind of like a tree-like structure. But maybe there's so much lateral gene transfer going on among bacteria that they would never find the real true tree because it doesn't exist. Okay? Right, so I'm going to leave you. So in a couple of hours, microbe of the week, right?